Maybe you're opening it on a phone.
So please, where should I open it?
No, but nonetheless, you not just after the zip folder, there are also a link. So there is also a link on your BS Science switch. You can search for staining. You type in staining and search. I think it should lead you to that.
Staining.
I think staining should be able to lead to that.
Okay.
Because I think processing, yeah, but staining should be able to lead you to the list of YouTube videos. Okay, it worked, right?
So I'm now searching for it.
Okay, so you can later you can let me know whether it worked or not. Okay, so we are looking at the muscle tissue, and in a presentation, this lecture, I'll give an overview of it and also comment on the types of muscle tissue. Now, this is one particular tissue that it's considered among the excitable cells in the body. It's considered among the excitable cells in the body. And the cells are specialized to increase in length. They can change in length. So they can lengthen and they can also shorten. And this muscle tissue is made up of cells that the primary cells, they are called myocytes. So whenever we are looking at the cells, the principal cells in a muscle tissue, we are looking at myocytes. And usually the cells are elongated in one direction. When they are occurring in groups, you find them oriented in one direction. So, but the key point is that these cells, they have the ability to increase in length and also reduce in length. Okay, and their name, the name of the cells is the myocytes. Now, the muscle tissue has important function in the body. One is that it's, sorry, today my pointer is not going to be, is the normal arrow. It's not going to be that red pointed structure. So just bear with me. Okay, so the muscle tissue has a lot of functions in the body. The first of this function that we can talk about has to do with movement. Well, movement, there are different types of movement. We have movement that, for example, involves moving from one place to the other, which is what we call the locomotion. It is a muscle tissue which plays an important role in locomotion. Also, we have movement that is sometimes you have various organs of our body and they may need to change in maybe position. Okay, so for example, the walls of the heart have to move from within a limited space of time. What that is in the course of in that limited space, limited space, they push on blood and move the blood throughout the body. So that is also a form of movement. If you look at our digestive tract, we swallow food and the food will be in our, it will land in our esophagus. But through the movement of the walls, the food that we swallowed will be able to move down the digestive tract all the way to the inner region. So all these are various types of movements which are actually mediated by which are performed by the muscle tissue. So then also in terms of stability, for you to be able to maintain your posture, all these are also as a result of the muscle tissue. Communication, I'm speaking now, I'm talking, and the talking is as a result. I'm able to produce sound because of muscles, the muscles in my voice box. Even my tank, the tank is also made up of muscles, and this muscles also help us in producing sound. You may have come across situations where some people's tongue were cut, and immediately it was cut. You notice that these individuals are unable to produce any form of sound. We also can also talk about the control of the body openings. The body opening. So you have, for example, the mouth. You can lip tight. You can actually lip tight your mouth, and no one can be able to force anything into your oral cavity. And we are able to do that because of muscles in our lips. Also in the inner region, sometimes, pardon me to use this, but maybe it may be necessary that maybe you may want to control your urine or control your feces. All these, sometimes you notice that at some point in time, you are able to control it. And now that amount of control that you are able to exert on this kind of body function is as a result of muscles in your body. Then also in heat production, your body may in times of cold, you know that you engage the shivering occurs, and the shivering is as a result of the muscles moving and moving your bones. Okay, bringing about clanging within your bones, and that is a way of generating energy in your body. So muscles tissue have a lot of functions in the body, and so that these are at least these are the functions that I'll talk about. Is there any clarification that I have to make? Okay. So the muscle cells or the muscle tissue have different properties. One of them is that they are excitable. They are excitable, and that they are able to respond to chemical, mechanical, and electrical stimulus. And muscles are considered as excitable cells. In our body, there are two excitable cells. We have muscle cells and the nervous cells. So that makes the muscle cells special. And they are able to initiate events that will lead to contraction. And the ability to initiate events that lead to contraction, we classify it under conductivity. They are able to cause the movement of ions, the transport of ions that will bring about contraction. And also, the third point has to do with the ability to shorten or lengthen, the change in the length. And that is what we call contractility. Then we also have what you call the extensibility. That is the ability to be able to stretch between these contractions. Then we also have elasticity. That is the ability to be able to return to their original length after stretching. So these are the properties of the muscle tissue.
Okay, Doc.
Please, any question?
Hello, hello, Doc.
Yes,
Hello.
please, I'm listening. Yeah.
Yes, Doc. Please, so with this, we are talking of the elasticity, that is the extensibility, the ability to stretch the muscles. Now, normally in sports, we have been hearing this word, the muscle pull. Somebody has gotten a muscle pull or something like that. Is it that there's a overstretch of the muscle?
Okay, so muscle pull has to do with, for example, when blood is unable to reach the muscles. So the kind of changes that they go through, the muscles will go through. When blood does not reach it, that is a sensation. But this, we are limiting ourselves to the cell. we are looking at the muscle cell. Then later we will extrapolate it to the muscle tissue. So maybe it is outside the scope of what we are discussing.
Yes, sir. Sir,
Yes.
I also think that when there is too much um, of the lactic acid, or when there is accumulation of lactic acid in the body, can also lead to muscle pull. Yeah. Just to add up to what you told.
Hello. Uh, Is it my network or is general?
I think that is off. I think it's general.
It's a general network.
Ah, why? Am I lost or what? Or the network is not working? Nicholas,
Please, you are not lost. Dog is off.
Nicholas, last time I called you back, I couldn't get you.
You mean me? When?
That's 3 days today.
Please, this class is being recorded.
But the lecture is out, so I'm not sure that.
The fact that the lecture is up doesn't mean you can have your conversation while the lectures are being recorded. Let's have some courtesy.
Madam, this is not anything. I was just telling him that I called him, I didn't get him. I'm not saying anything bad.
Hello? You have to mute your mic before you communicate to somebody else. Martha.
Yeah, please, sorry, I went offline. I'm very sorry. Okay. Sorry, I think we were talking about the muscle pool. And Well, someone suggested drew attention to the buildup of lactic acid. Yes, and that occurs during anaerobic respiration. Okay. Sorry, let's continue. So when
Doc, please, I have a question.
Okay.
I want to ask, you see, when you go to gym, after exercising the body, you could see that the muscles will expand, but when you get home for some time, it will later contract. I want to know the cause of that.
The muscles expanding.
Yes, it will expand after the exercise when you go to gym. But as soon as you get home for some time, you see the body will come back to its normal position after the expansion of the muscles.
I think that's the question.
Okay, I have not paid attention to that. What I know is that usually, when you exercise the muscles for a longer time, then you can see the hypertrophy of the muscle. The muscles become bigger. They become more developed. But within that short time, per adventure, like maybe what I can think of is blood flow. But I think someone was also making a comment. So let's hear from the person. Please say any other suggestion. Have I gone offline? Okay, so we have as a muscle cell, the muscle cell has the characteristics of like the eukaryotic cell, just that it has names and which we may have to adapt. So I will take you through the parts of the muscle cell and the names that are ascribed to it. So first, we have what you call every cell has a plasma membrane, including the muscle cell. So the muscle cell, like any other cell, will have a plasma membrane. Eukaryotic cell has a plasma membrane. And the plasma membrane of a muscle cell is called sarcolemma. Sarcolemma. Then the muscle cell has a cytoplasm, and it is also referred to as a sarcoplasm. Sarcoplasm. And one thing about the cytoplasm of muscle cell is that it contains an organelle called glycosomes. And it is in these organelles that they store glycogen. They store glycogen because the body stores excess glucose in the form of glycogen. Then we have myoglobin, which is an oxygen binding pigment. We are used to hemoglobin, which is present in the red blood cell, but also the muscles also have a pigment that is able to bind to oxygen, and we call it the myoglobin. Also, the muscle cell has an endoplasmic reticulum, but and its name is preferably called the sarcoplasmic reticulum. Then in a muscle cell, if you pay attention to the pointer, the muscle cell is able to increase in length and decrease in length. But this increase in length and decrease in length is attributed to some protein fibers, which serve as the contractile elements in the muscle. So and these are what we refer to as the myofibrils. So the myofibrils are the contractile element in the muscle cell. Then for us to be able to study this muscle cell, the whole length, the myofibrils is relatively longer, but to be able to study the muscle cell, we have divided the myofibrils into segments. And when we study that particular segment refers to the contractile unit of the muscle cell, and we refer to that segment as the sarcomere. the sarcomere. So the contractile unit of a muscle cell is referred to as a sarcomere. Now, having considered the parts of a muscle cell, we look at the types of muscle tissue. So there are 3 types of muscles in the body. And we have a muscle that is, with the more common one is the muscles that are attached to bones. We call it a skeletal muscle. Many of us have gone to the meat shop to buy meat, and you notice that there are the red meat that we usually buy, they are attached to bones. And those, what we consider as red meat, is actually a muscle. But these muscles are called skeletal muscles because of their attachment to bones and cartilages. Then we also have another type of muscles which is present in the heart, and that is what we call the cardiac muscle. It's exclusively in the heart, the cardiac muscle. Then there is a third form of muscle which is present in our digestive tract and other internal organs of our body. And these are what we call the smooth muscles. So we look at the different types of these muscles, and we'll discuss them. Now, this is a schema or a micrograph of how the 3 muscles look like. So we have, they differ under the microscope. And
so sir, hello sir.
Yes.
Please concerning the cardiac muscle and then the smooth muscle, do they also have the contractile tissue in it?
Yes, especially for skeletal. Later you notice that skeletal and cardiac, it is clear, but the smooth muscle, it varies slightly.
Okay. Thank you, sir.
Okay. These are pictures. These are the different types of muscles that you and this is a schema of how they look like and under the microscope. But let's take the muscle one after the other. So we have, for example, the skeletal muscle. We have the skeletal muscle, and this skeletal muscle is relatively is elongated. See, it is long and cylindrical in shape. And this is the entire cell. You have to look at it as it's a cell. And because it is elongated, usually we use the word fiber for it. The skeletal muscle cell is multi-nucleated. So it has several nuclei within this cell. This cell is multi-nucleated. And the cell is having a striped appearance. So just like we see in the zebra, the skeletal muscle cell has this striped appearance, and therefore we use the word striated. So we see that the skeletal muscle is striated. When you look at the body, it is made up of alternating light and dark bands. So alternating light and dark bands give it a striated appearance. And the skeletal muscle is under voluntary control. This is a muscle that you can actually control. You can decide to, you can initiate when you feel like, okay, I want to perform this kind of activity. It may be, so long as you decide, then the body would go on to activate this muscle. The contraction, so these are characteristics of the skeletal muscle tissue. And the next thing is to comment on what the myofibril in the skeletal muscle cell. So I indicated that the skeletal muscle cell is able to increase in length and also decrease in length. And it is as a result of the myofibrils which are present. Now, when we look at the myofibrils, we see that the myofibrils is made up of a region which is dark and light. So there is a segment that is a dark band, so we refer to that segment as dark band. And there is a region that is light colored. We refer to it as a light band. So on this slide, you see that this is the myofibrils in the skeletal muscle cell. This is one myofibril. And you notice that the myofibril, there is a dark region, a light region, a dark region, a light region. Now, and even with the light region, you notice that one of them has a zigzag, a zigzag line running in between. Actually, the light region that is having the zigzag line, that is our light band. That is the light band of the myofibril. Then we have the dark region with also the light band in between, the lighter region in between. The entire thing is the dark band. Okay, so this refers to our dark band, and we call the dark band the A band. The A in the dark, when you are spelling dark, the letter A should help us remember the A band. And when you are spelling, when you look at the word light, the I in the light should help us to remember the I band. So if you look at the structure of the myofibril, it has a dark band, which is referred to as the A band. And even the A band has in it, there is a light region, which we call the H zone. There is a light region in it, which we call the H zone. Now, after the dark band, we have the light band or the I band. And the I band is bisected by a line which we call the Z disc. The Z disc. Then after the Z disc, you see that there is also another A band, which is a dark band with this H zone, followed by another I band with a Z disc. Now, in the structure of a myofibril, the section of the myofibril between two Z discs, so between these two Z discs, this is one Z disc, and that is the other Z disc. We call it sarcomere. Sarcomere, and this is what constitutes the contractile unit of a muscle fiber, the contracting unit of a muscle fiber. Also associated with the myofibril is the endoplasmic reticulum, that is the sarcoplasmic reticulum. And this sarcoplasmic reticulum is positioned at the junction between the dark band and the light band. So we call it the AI junction. So that is an interesting part of the sarcoplasmic reticulum that is associated with the myofibrils. So the sarcoplasmic reticulum in a skeletal muscle has regions which are more horizontal and the ends are expanded. It has horizontal regions, but when you get to the ends, it is expanded, and we call that expanded region terminal cisterna. Terminal cisterna. Now, in this terminal cisterna, you have two terminal cisterna lying side by side to extract a modification of the plasma membrane. We call it the T-tubers. So there is a part of the cell membrane which has invaginated into the cytoplasm, and we call that invaginated portion of the plasma membrane T-tubers. Now, occurring on each side of the T-tubers is a portion of the endoplasmic reticulum, which we call the terminal cisterna. Now, the terminal cisterna, the two terminal cisterna The two terminal cisterna and the T tubers constitute what we call the triad. It constitutes what we call the triad, and this occurs at the junction between the dark band and the light band. So we refer to this region as the AI junction. Please, is there any question or any clarification that I have to make on this slide? Are we clear with the information?
There are some terms that you mentioned which are not seen in the diagram.
Okay, so which terms?
Terms like 69 and something else.
Okay. So I think may have to, I'll update the slide to reflect that.
Okay, thank you.
So we have a zone in the middle of the A band. Okay. And so I mentioned the T tubers, which is so we have the terminal cisternae. Okay. Which is a section of the smooth endoplasmic reticulum that is present at the AI junction. Then we also have the T tubers. Which is an invagination. Of the plasma membrane. Then I think I also mentioned I think I mentioned another term, which is the triad. So I think I'll update that. So the T tubers plus the two terminal cisternae. Okay, is it better now?
Yes, sir, but please, the teachable, the imagination. I think
Invagination.
Invagination.
So
the spelling you omit some.
Invagination, yeah. Not imagination, invagination. So you have the plasma membrane, and the plasma membrane is actually extending inward into the cytoplasm of the skeletal muscle cell. So that is the invagination.
Okay, sir. Thank you, sir.
Okay. Now we go on to look at the detailed structure of the myofibrils. The myofibrils are made up of thick and thin filaments. The myofibrils are made up of thick filaments, and the thick filaments are colored orange, and the thin filaments are colored blue. Now, the thick filaments are made up of, it's actually made up of a molecule called myosin. Myosin. So the thick filaments are made up of myosin molecules. Now, myosin has, when we look at the structure of the myosin, it has a structure, it's similar to the golf club, the club that we use to play golf. So it has a head then it has a tail. So it has a head and a tail. So that is one characteristic of the myosin molecule. Then on its head, it has a part that allows for the binding of ATP. So it has an ATP binding site on its head. And when we talk about ATP, that is the currency of energy, and it is the adenosine triphosphate. So on the head of the myosin is an ATP binding site. When we come to the thin filament, the thin filament is formed, it appears as two strands that have been twisted around each other. So it's like two strands of proteins that have been twisted around each other. And this, the twist is actually a polymerization of the actin molecule. So it is actin molecules which have been joined end to end to form a strand. So you have two strands of actin molecules, and the actin molecules are the strands have been twisted around each other. So this is a picture representing the actin molecules, and when they have been arranged end to end, they form individual, they form a strand, and the strands have been twisted around each other. Also occurring together with the alongside together with the actin molecules in the thin filament are other proteins. So we can talk about other proteins like tropomyosin and troponin. So we can talk about other proteins that occur side by side to it, and that is tropomyosin and troponin. So we have tropomyosin, and troponin. So these are proteins that occur side by side. So troponin and occur. So so tropomyosin and troponin are present in the thin filament. Then here also I want to add that the head, I think I mentioned that it has a site for ATP to bind. Okay. So this is, I've talked about the skeletal muscle cell, what is in the cytoplasm, especially the myofibrils, and I've indicated what the myofibrils they look like, and also the detailed structure of the myofibril. Okay, next, I want to draw your attention to the fact that when you go to the meat shop to look at and we see the meat, what is seen as a meat is actually an arrangement of these skeletal muscle cells, which a higher organization of the skeletal muscle cell. And this is and this organization is achieved because of association of connective tissue. So the skeletal muscle cells are supported by connective tissue framework, and there are different levels of connective tissues which are present in them. And I would like us to know the connective tissues. So on the slide, you have a bone, and the bone has a muscle attached to it. That's the meat, which is attached to it. When we do a further breakdown of the muscles in the muscle cells in the meat, we would start with, we come back to this kind of level. So you have a skeletal muscle. This is a skeletal muscle cell or a skeletal muscle fiber, and occurring within it are the myofibrils. Now there is a loose connective tissue that surrounds each skeletal muscle fiber. And the connective tissue, the loose connective tissue that surrounds each skeletal muscle fiber or skeletal muscle cell is what we call the endomysium. Endomysium. And it is an areolar connective tissue. Now, beyond the skeletal muscle cell and its endomysium, you have a number of them coming together. So you have a number of skeletal muscle cells with their surrounding immediate endomysium occurring in a bundle. So they occur in a bundle which is called fascicle. And even at that level, there is a connective tissue that surrounds the entire fascicle. The name of the connective tissue that surrounds the fascicle is the perimysium. Perimysium. Then beyond the fascicles and its associated perimysium, you have a number of these fascicles occurring together, and they are all surrounded by their own perimysium. Now, this composite is also now surrounded by a thicker or a dense connective tissue called the epimysium. So the epimysium surrounds the several fascicles which are present in a skeletal muscle cell, whereas the fascicle is actually a composite of several skeletal muscle cells and their associated endomysium. The epimysium extends distally, extends beyond the fleshy muscles and become twist-like, like a rope, and attach to the bone. And the name of that extension of the epimysium that attaches to the bone is what we call the tendon. So you often hear that muscles are attached to bones via tendon. Please, any question?
So please, if you can repeat the last statement you made regarding the tendons. You said the extension of the epimysium.
Epimysium beyond the muscle cell. So the muscle cells are relatively fleshy, but when the part of the epimysium that extends beyond that goes beyond the skeletal muscle cell, they become twist-like. Okay, they become twist-like and they attach to the bone. And the name of that segment of the epimysium that becomes a twist-like structure and attaches to the bone, we refer to it as a tendon. So many times in books you hear that muscles are attached to bones by what structure? They attach to bones by a tendon. Please, is that point, is it a little clearer?
Yes, please. Thank you.
Okay.
Yes, please.
Yes. Okay. So I've also added, there is on the slide, we also have another picture which illustrates the connective tissue organization. So you have a muscle fiber, and the muscle fiber is surrounded by the endomysium, which is this white region around the muscle fiber. Then several muscle fibers and their respective endomysium are surrounded by another layer of connective tissue, which we call the fascicle or bundle. Then you have several fascicles or bundles also surrounded by a thicker connective tissue called the epimysium. The epimysium. In a body, there are two types of skeletal muscle fibers. We have red and white skeletal muscle fibers, and that's the red and white skeletal muscle cells. Often the red muscle cells are comparatively thin, but they contain large amount of myoglobin and mitochondria. When a cell contains a lot of mitochondria, what happens is that it means that it is highly dependent on oxygen. So it will require more oxygen so that it can burn and generate ATP for the body. Then it also contains myoglobin. So the myoglobin is an adaptation of the red skeletal muscle, in the sense that the myoglobin binds to, is the oxygen pigment in the skeletal muscle, in the muscle, and it binds to oxygen. Often the red muscle cells, it comes for them to contract, contraction has to do with the muscle trying to reduce in length. That is, if they shorten. That shortening of the red muscle cell is relatively slower when it comes, when compared to the white skeletal muscle cells. And this is attributed to the fact that the red muscle cells contain an isoform of myosin with a very low ATPase activity. That is, its ability to, the rate at which it uses the ATP is relatively slower, and therefore the red muscle fiber ends up contracting slower. Red muscles are used when there is a need for you to be able to put for an activity which requires that you sustain production of a force. Okay, you produce a force, but that force needs to be sustained, and that is where the red muscles are often used in the body for. Like, for example, you are standing for a longer time, and it requires that you maintain posture. For you to be able to maintain posture, you notice that it is more like a 400-meter race. Okay, it needs time, so you need to generate that kind of force in order to be able to achieve that. I want to reorganize this one and make it better. So I think it's supposed to be under this to make the information more meaningful. So I said that the myosin head, for example, has a site that will allow for ATP to bind. But if you look at when the ATP binds, the myosin head has an enzyme which we call the ATPase enzyme. And the ATPase is able to break down the ATP to generate energy for their activity. So the contraction in a red muscle cell is relatively slow because the myosin that is present, that it has a slower ATPase activity. The white muscle cells are thicker and they contain less myoglobin. The ATPase activity of the white skeletal muscle fibers is high, and therefore contraction is relatively fast. Please, any question on the skeletal muscle? Okay, then we look at the cardiac muscle. So the cardiac muscle is also elongated like the skeletal muscle. However, it is different from the skeletal muscle in the sense that the skeletal muscle is multinucleated, but the cardiac muscle is mononucleated, or you can say uninucleated. It has one nucleus. And even with the nucleus in the cardiac muscle is positioned at the center. But in that of the skeletal muscle, you find that the nucleus is located at the periphery. Did I say it? Oh, this is my first time. The nuclei in the skeletal muscles are located at the periphery, but that of a cardiac muscle is located at the center. So this is the cell, the cardiac muscle cell, and the nucleus is located at the center. The cardiac muscle cell itself is also, it has this striated appearance like the skeletal muscle. So when you look at it, it is seen as alternating, it appears as alternating dark and light bands. Another difference, there is an interesting feature of the cardiac muscle, and the cardiac muscle is branched. So at a point in time, the cardiac muscle cell splits. It splits. And when it splits, each split will form a junction with another cardiac muscle cell, or with another part of another cardiac muscle cell. Now, the parts that the split ends of the cardiac muscle cell meet other cardiac muscle cell is what we call the intercalated disc. So the cells meet one another, or they become contact with one another at the intercalated disc. There are two types of cardiac muscle cells. We have what we call the contractile cardiomyocytes, and we have the pacemaker cells. The contractile cardiomyocytes, they generate the force with which the heart is able to contract and able to move blood into circulation. So the contractile cardiomyocytes, when the heart is pumping, the cardiac muscle cells are contracting in order to be able to move blood, generate a force, and move blood through a particular distance. But these contractile cardiomyocytes, so that is where the force with which the heart generates in order to move blood, that force is generated by the contractile cardiomyocytes. Then we have another group of cells called the pacemaker cells. The pacemaker cells in the cardiac, these are actually responsible for the autorhythmic activity in the heart. The heart on its own is able to beat. The heart on its own is able to beat. It doesn't need to be connected to the body. When we decouple the heart and we put it in a physiological condition, you will see that the walls of the heart will be able to beat on its own. And that ability of the heart to beat on its own is attributed to the pacemaker cells. Now, these pacemaker cells have the ability to be able to generate electrical activity within the heart. It is the electrical activity that it generates within the heart that brings about the contraction of also the contractile cardiomyocytes. In the heart, there are different pacemaker cells. So we have those that we call the SA node. We have another pacemaker cell that we call the AV node. Another group of pacemaker cells, which we call the common, we call it the bundle of His. Another that we call the branches of the bundle of His. Then the final one, we call it the Purkinje fibers. They are the different pacemaker cells in the heart. So we have the pacemaker cells in the SA node region, pacemaker cells in the AV node region, pacemaker cells in the bundle of His region, pacemaker cells in the bundle branches, and the pacemaker cells in the, which are the Purkinje fibers. I think what I will do now is to add the names of the location of the pacemaker cells on the slide. Then we look at the smooth muscle cell. The smooth muscle cell is quite different from the skeletal and the cardiac. So the cell is having a spindle shape. It is fusiform in shape, but it doesn't have. It is not striated. You don't see.
Hello.
You don't see the alternating dark and light. Hello.
Yeah.
Please, is this a question? Okay. So the smooth muscle. Yeah, who is speaking?
Please carry on.
Okay. So the smooth muscle cell is fusiform in shape. I think someone has person should mute himself.
Hey, wisdom.
Hello.
Yeah, the skeletal muscle cell is fusiform in shape. It has a spindle. It has a spindle shape, and it also has a nucleus. But one nucleus, but the nucleus is at the center of the cell. You don't see the alternating dark and light bands on the walls of the smooth muscle cell. So you say that there are no cross striations, or you can say that a smooth muscle cell is not striated. So because it doesn't have the striation, the sarcomere relationship of myosin to actin, which occurs in the skeletal and cardiac, does not exist. The smooth muscle cell, just like the cardiac muscle cell, is not under your control. It is involuntary. And we have to bear in mind that though the features are different, it does not make the smooth muscle cell anything lesser. The smooth muscle cell is as strong as the skeletal muscle cell. And one thing about the smooth muscle cell is that it is highly resistant to fatigue. Unlike skeletal muscle cell, you notice that when you go to the gym and you are lifting a dumbbell, those muscles in your arm are skeletal muscle cells. But when you lift the dumbbell for a longer time, it gets to a time that you get tired. It's not like you have the will to continue, but the muscles are tired. The muscles are tired and they will not be able to perform those functions. Now, this is where we talk about fatiguing. Smooth muscle cells don't do that. So smooth muscle cells don't get fatigue. However, the cardiac and the skeletal muscle cell rather gets fatigue. On the slide is a presentation of the smooth muscle cells. So you can see the nucleus, which is oval shaped. You can also see the part that is not well defined. That is the cytoplasm of the cells. And in between, you find some blood vessels, which are also running through the smooth muscle cell to supply the cells. When it comes to the smooth muscle, the sarcomere relationship does not exist. However, there is a homologue of the myosin and also the actin. And this will let me introduce you to the concept of dense bodies. Dense bodies are structures that are scattered throughout the cytoplasm of the smooth muscle cell. And also, it can also be attached to the wall of the smooth muscle cell. The dense bodies function as the Z disc. The dense bodies function as a Z disc. And when it functions as a Z disc, actin molecules would attach to it. So you have thin filaments attached to the Z disc. And you now have the thin filaments interacting with the thick filaments in the cytoplasm in order to bring about movement. In the body, the smooth muscles, yes.
The picture that just the top one is that one that the Z disco.
No, the dense bodies. So these are the dense bodies, the black. The black circles that you see in the picture represent the dense bodies. They represent the dense body. So, yeah.
Okay, thank you, sir.
Okay. In the body, the smooth muscle cells are organized in bundles, which we call they are sheets. They are organized in bundles. And these, whilst they are organized in bundles, they cause a particular orientation. So some you find, this is a picture of the smooth muscle, different smooth muscle. If you look at it, you can see the fusiform shape. The nucleus is at the center, and it is just one nucleus. Then you can also pay attention to this part. So you see that it's oval in shape. The cytoplasm is there, and we have the nucleus. Now, when these smooth muscle cells occur together, depending on how they are oriented, then they will tell us, depending on, hello?
Yeah, please, when you look at the structure, the circular smooth muscle, you realize that it has a hole. Does it mean that they consist of lumen? Can we say they consist of?
I think, okay, I'll come to that. I'll comment on that. I'll comment on that. Okay.
Okay. All right.
So in organs like stomach and then digestive tract, the muscles within this are arranged in a particular pattern. So some, the common one, sometimes some of the fibers are oriented. They go around they are arranged circumferentially around the organ. So just like a cylinder, the curved part of the cylinder, you have the fibers being arranged so that they go around the curved part of the cylinder or the gut. Now, in this picture, you have the digestive tract. This is a digestive tract, and the digestive tract has an opening at the center. This is the lumen through which food will be passing. Now, within the walls of the digestive tract, we have a wall which is made up of muscles. And the muscles are usually in most parts of the digestive tract. The muscles in the inner aspect, which is closer to the lumen, are arranged circumferentially around the muscle. So they are arranged in this pattern. And the muscles which are found on the outermost layer, they are arranged parallel to the long axis of the muscle. So we use a cliche, inner circular, outer longitudinal. So the outer longitudinal muscles are arranged such that they run parallel to the long axis of the digestive tract. And in these muscles, contraction is usually slow and may take even minutes to develop, unlike skeletal muscles. But when they get their contraction, can sustain it for a relatively long time. Where can we find the smooth muscles? So we can locate muscles, smooth muscles, in the walls of viscera, the whole organs, like the stomach. The stomach, where when we swallow food, the food will land in the stomach. The uterus, the ureter, blood vessels, even the eye, the stomach, the abdomen, there are hello? Hello.
Yes. Yes, sir. We are here.
So you can locate the smooth muscles in the walls of visceral hollow organs like the stomach, the uterus, ureter, blood vessels, and even the eye. And usually they are under involuntary control. So you cannot control your smooth muscles. Now it is worth noting to
Sir,
yes.
concern about the involuntary action of the smooth muscle.
Yes.
Assuming someone is having a big tummy, I want it to be flat and start with a stomach training. With time and the stomach
No,
reduces or something.
yeah, so we should be cautious of the word stomach. Stomach is an organ in the abdominal cavity. So I think what they usually want to refer to is that when you are doing some abdominal exercises, so that we can disease. Stomach is an organ in your abdominal cavity. So where the question has to do with more of abdominal muscles. Okay, so this is related to the makeup of our abdominal wall. In the makeup of the abdominal wall, yes, there is a section for muscles, but these muscles are skeletal muscles. Now, what gives us a pot belly is as a result of accumulation of fat. It's accumulation of fat. So fat, when fat lies between the skin and the muscle, then it presents as a pot belly. If we want to reduce the pot belly, it comes with a number of conditions. So one is what is allowing the fat to accumulate in the skeletal muscle. So the fat is accumulating because you are taking in energy, which is way more than what you need. So because it is way more than what you need, the body will have to store the excess energy in the form of fat. So, exercising is one way of burning the fat. And secondly, we even encourage people to also stop taking in excess energy so that at least they can provide what is necessary for their body.
So, what about the case of diarrhea? I know it has to do with some loose contraction of the muscles. If you can expatiate on that.
Okay, so when we say diarrhea, diarrhea is watery stool. It's watery stool. Now, when the food is passing through the digestive tract, the digestive system will add enzymes and also digestive enzymes in solution. They will add it to it. The body has a responsibility of absorbing back the fluid that they lost. However, in situations where that fluid is not absorbed, then you have a lot of fluid occurring with the feces, and that will bring about the diarrhea. So the fluid, if the body does not absorb most of the fluid, then you end up with having a watery stool. Now, additionally, many of these diarrhea occurs with infection. It also occurs with infection. So during the infection, it can bring about contraction of the smooth muscles at a relatively faster pace, and that will also promote emptying of your like you having this frequent urge to go and attend to nature's call. I don't know whether the explanation is a little clear.
So it's clear. Thank you.
Okay. So on this slide, I want us to look at compare and contrast the various muscle tissues. So we have, for example, this is a longitudinal section of the muscles. And if you look at it, you notice that the skeletal muscle and the cardiac muscle have this alternating dark and light bands, but the smooth muscle does not have it. So that is one difference. So you can say that the skeletal muscle is striated, cardiac muscle is striated, but the smooth muscle is not striated. Also, we look at the nucleus. The skeletal muscle has just one nucleus, has multiple nucleus, and they are located at the periphery. But if you look at the cardiac and the smooth, you notice that each cell has just one nucleus, and it is centrally placed in the cell. Also, in terms of our control, skeletal muscles and cardiac muscles are under control. But in the case of the smooth muscle, it is not under your control. It is. We can also pay attention to the transverse section. We can also pay attention to the transverse section of the skeletal of the muscles. So here in the transverse section, you see that it's like a circle which you are cutting. You may not get a perfect circle. Nonetheless, let's look at it. So here you see that the nuclei are at the periphery. The nuclei are at the close to the plasma membrane. So all of them. So this is why we call that. This is why we say that the nuclei of a skeletal muscle cell are located at the periphery. Now, when we come to the smooth muscle cell, the nucleus is at the center. The nucleus is at the center, and it is only one. Okay. Here you in the skeletal muscle, you can see more than one. Then we come to the cardiac muscle. So it is only one nucleus, and it is also at the center. And the interesting thing is that smooth and cardiac muscle are not under your voluntary control. Okay, so this will bring us to the end of the lecture. You may want to do some, if in case you want to do additional reading, we've added some information that you can add. But at least in this lecture, you should be able to tell us, we've talked about the structure of the skeletal muscle cell, what is in the cytoplasm, what you are supposed to look out for. We've also talked about the striated muscles, the cardiac and the skeletal muscle, what you are supposed to expect in the cytoplasm. Okay, the myofibrils and what is the structure? They are made up of alternating dark and light bands. So these are some of the things that you should look out for. Please, any question?
Sir.
Yes.
Please, can we get some of the tutorial questions?
Yeah, I think Madam Rebecca has shared it in your group. So when the lecture started, some of your colleagues raised that issue. So I worked on it and I've sent it to. So okay. You have those questions in your group. Okay. Okay. Sorry for the hitches in the internet connection, but I think we will take a break and resume in 30 minutes. What time? Maybe it's. Let's continue at 9 o'clock.
It's 8:30 now.
So we continue at 9 o'clock.
Okay, sir.
Okay, thank you very much. I'm working on my internet connection.
Hello, sir.
Okay. Yes.
Yeah, so you see that sometimes because of internet connection, some of us we don't really follow directly the lectures. Maybe you have to watch it after the lesson. So sometimes like the abbreviation, the maybe and other things. Sometimes you have to give us the full meaning of those things, because when you are watching the video after this thing, you can't have the opportunity to ask questions and know which of what do you mean.
Okay, so are you requesting?
The question again.
Yeah, what you are requesting?
I'm requesting that it's like, sometimes some of the names, instead of you to write a full name, just use maybe CC, VA, and those things, those abbreviations, to mean maybe the short form of writing certain things.
Okay.
So because sometimes, because of internet, you have to watch the video after the lesson then you have to make it like give her the full meaning of those things so that we not struggle understanding it when we are reading the slide.
Okay. 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 So we'll look at that. Okay, so let's meet at 9:30 and continue. Sorry, 9 o'clock and continue. Okay, thank you.
I say good morning, colleagues. But you wanna tutorial questions in one fan to my
Anope was out to move, was out to. Did go back to the chat. Men nationwide or the nationwide.
But me that.
Yeah.
Okay, so I think we can continue. So we are looking at the nervous tissue, and I'm hoping that at the end of the lecture, we will be able to state the functions of a nervous tissue. You should also state the functions of the different cells in a nervous tissue. You should be able to describe how information is conducted along axons and across synapse. So these are our lecture objectives. Okay. So we would let's go on with it. So this will be the outline of the presentation. But first, I'll give an overview of the nervous tissue. So the nervous tissue is actually what is present in the nervous system, the organs of the nervous system. And the nervous system it helps us to be able to perceive changes in our internal and external environment. And usually the changes in the environment is what we call the sensation. So in biology, sensations, you being able to sense changes in your environment, you classify them as sensations. Now, the things that we change, the changes that we experience in our environment, some of them are special. Usually, those which are considered as special are restricted to a particular part of our body, but the general is experienced by multiple parts of our body. So, for example, pain, you can experience pain. Yeah, you experience pain in your body, your skin, it can be your face, your back, even sometimes in your oral cavity, you can experience the pain. So it is not restricted to a particular body function, and hence is considered as general. Then we also have the ability, temperature changes. So whether the place is hot or cold, you notice that you can experience in different places. So that is not also special. We have even situations where people can actually put food in their mouth and they can experience it, whether the food is either hot or cold. Then touch, you can be touched in different places. Pressure also is also possible in different places. But let's look at sight. Let's look at sight. So when immediately we talk about sight, we are referring to the eyes. We are referring to the eyes. If we also talk about taste, taste will restrict you to the tongue. What about audition, hearing, sound? Sound will always bring our attention to the ear. Smell, that is the nose, and balance is still part of the ear. So these kind of sensations are actually special because they are restricted to a particular part of our body. The other function of the nervous tissue is to enable us to elicit appropriate responses to the sensations, the changes in sensation. So, for example, you walk barefooted in a place, or you are in a place and accidentally you step on a hot object. You step on a hot object. Your body, your nervous tissue will help you to note that there are changes in the environment. That is in terms of the heat. And the appropriate response will be for you to be able to jump. You jump over and you jump onto a safe place. And that is the appropriate response. You may be looking at let's say you go to a room that is hot, and all of a sudden you notice that you started sweating. So the response will be the sweating, but the changes in sensation will be the temperature. So all these are activities of the nervous tissue. Now, I use instances where you step on a hot coal, you jump, you move into a hot area, and like you sweat. All these kind of responses are they boil down to three main structures, like four main structures, which include skeletal muscles. So the muscles that are able to move our bones, the cardiac muscle, that is your heart. You find yourself in an uncomfortable situation, and your heart has to be beating faster. Also, I use the issue about sweating, and the sweat is produced by the glands of our body. So we looked at the exocrine glands and the endocrine glands. We are referring to the exocrine glands. So the exocrine glands would also be involved so far as the nervous system is giving the appropriate feedback to the people. Last one is the nervous tissue even is involved in memory. For you to be able to remember something is the nervous tissue, so it is also involved in memory, and maybe you can note it down. Okay. Now, whenever we come to the nervous tissue, the nervous tissue usually comprises of two types of cells. We have the main cell, which is the neuron. Then we have the supporting cells, which we call the neuroglia. The neuroglia. Others even use the word glia. So these are the two main types of cells at present in the nervous tissue at any point in time. There are functional differences between the neurons and the neuroglia in the sense that neuroglia can still divide freely compared to the neurons. Neurons don't divide. They don't undergo cell division. Even if they occur, it takes a longer time. Okay, so the neuroglia are supporting cells in the nervous system, and they can divide faster, where neurons are not. They can also transmit. They can, but one thing is that they do not transmit nerve impulses, of which they do not transmit nerve impulse, which is a main function of the neuron. So these are the components of a nervous tissue: a neuron and the supporting cell. I want to comment on the location of the nervous tissue. So
okay, sir, please, I have a question.
Okay.
Okay, sir, please, my question is, how does or is the nervous tissue able to send information to the brain for interpretation?
Okay, so it's actually outside our scope, but can I finish that slide? I'll add it. Next slide, I'll add it.
Okay, sir.
Okay. So the nervous tissue can also be described based on the location where it was taken. So, and this will bring us to the components of the nervous system. So, for example, the nervous system is made up of, we can divide the nervous system into two: the central nervous system, which comprises of the brain and the spinal cord. Then we have the peripheral nervous system, which consists of the cranial nerves, which are nerves attached to the brain. Then we have spinal nerves, which are nerves attached to the spinal cord. Then we have some swellings, nervous tissue swellings, which we call ganglia. So if so in the course of the lecture, I'll be mentioning some cells, and if I mention central nervous system, please bear in mind that I'm referring to the brain and the spinal cord. If I'm talking about the peripheral nervous system, I'm talking about the cranial nerves, spinal nerves, and the ganglia. Now back to how the brain is able to receive information from a part of the body. So you see, nerves are connected to the brain, either the brain or spinal cord. So, and these nerves are linked to your body parts. So whenever any part of your body is maybe triggered, okay, the information flows through the nerves and enter the spinal cord. So when they enter the spinal cord, in the white matter of the spinal cord, the information can rise up the spinal cord and move towards the brain for interpretation. I don't know whether you this description is a little okay for you.
Yes, sir.
Okay. It's that is also another entirely different topic on its own. It doesn't fit in our scope. Okay.
Okay, sir.
So we have we'll talk about supporting cells of the nervous system, and we have a number of them. So we have the ependymal cells, we have astrocytes, we have oligodendrocytes, we have microglia, we have Schwann cells, and we also have the satellite cells. So we'll take them one by one and explain. So ependymal cells are the nuclear. Hello.
So my hand is up.
Okay.
Yeah, when on the slide number 7, there is neuroglia are responsible for the brain tumors. And with
Yes.
that, I don't know, is it the function or the malfunctioning aspect of the neuroglia that is responsible for the tumor?
Okay. So it is the abnormal functioning. Is abnormal functioning of the neuroglia. So perhaps it is better to put it in this way. They are implicated in brain tumors.
Okay. Okay, sir.
So often, when it comes to tumors in the brain, it mostly involves the neuroglia. Okay. So look at the different types of neuroglia, and first is the ependymal. So the ependymal cells, they are present in the brain and spinal cord. Now, in the brain and spinal cord specifically, there is a cavity. They have the brain is not a solid structure. There are also cavities within the brain through which cerebrospinal fluid can flow. And in these cavities, they are lined by an epithelium. And the epithelium that lines the cavities of the brain and also that of the spinal cord. So it's actually the ependymal cells. So these are the ependymal cells. So they are serving as epithelial. They are the epithelial lining of the in the brain of the in the brain cavity and leg. Okay, so that is a picture. This is the ependymal cells. This is the ependymal cells. You can see their stereocilia. With this stereocilia, they beat in order to move CSF. The second of the cells, that's the astrocytes. So astrocytes derive their name from the fact that they are star-shaped, and they are very important in forming the blood-brain barrier. So in our body, the brain it's not every structure, everything that should cross into the brain. The body highly select them. Okay. And in order to be able to do them, it requires that it puts up a barrier that will not allow every substance to just pass through. Okay, and that is what we call the blood-brain barrier. So we have the blood, then we have the brain, and we have the barrier. Okay, so the blood-brain barrier is very, very important. If it's broken, what it means is that blood will mix with the cerebrospinal fluid, and the nervous tissue is such that blood should not come into contact with these cells. Okay. The next cell is the oligodendrocyte. So oligodendrocyte.
Hello.
So yes.
Please, with the blood-brain barrier.
Yes.
You said if it breaks down, blood will mix with the.
It will come into contact with the CSF.
Okay, so if that happens, what are some of the conditions that a person will get in case something like that happens? Or what are the dangers?
Well, the dangers is the commonest is a stroke. Because when blood comes into contact with nervous tissue, then the cells will die. And when that part of the cell dies, that the body part, the function of a body part will be lost. And that is where stroke comes in. I don't know whether it's a little clear.
Hello, sir.
Okay.
Yes.
Hello, sir. So in that manner, how does it receive nutrients, oxygen, and then nutrients? That nerve, if it doesn't come in contact with blood, then how does it receive nutrients and oxygen?
No, the brain and the brain does not, the brain tissue, the neurons and they don't come into contact with blood. So the question is, how do they receive nutrients?
Yes, sir.
Please, is that okay? So
yes, sir.
At the blood-brain barrier, it's selective. Not all substances will pass through, but nutrients, a number of the nutrients like oxygen, they pass through.
Okay.
Okay. We have the oligodendrocytes, and these are also present in the brain and spinal cord. But what their function is that they are able to they produce myelin sheath, which is a fatty substance. It's a fatty substance that is used to cover the axons of neurons. So it's the oligodendrocyte that produces that myelin sheath. And it will interest you that when the myelin sheath is present, it decreases the time. It decreases the time that will require for information to be transferred throughout the nervous system. Okay. The next is yes.
Sir, please take that point again. You said it does what?
It lays myelin sheath, and when the myelin sheath is present in a neuron has a myelin sheath, it increases the rate at which information is transferred by the neuron to the brain and spinal cord. Is it a little clear?
Yes, sir.
Okay. Then we have the myelin sheath, but you see, the myelin sheath is formed in the peripheral nervous system. They are formed in the peripheral. It's formed in the peripheral. They form myelin sheath in the peripheral nervous system. So when you have a nerve and a ganglion being surrounded by myelin sheath, then it's because then you are looking at structures. These structures are in the peripheral nervous system. Okay. We also have another cell, which is the microglia. This is present in the brain and spinal cord. And their function is to dispose of debris. If there are any foreign particles in the nervous system, be in the brain, being it normal and abnormal, it is the microglia which will go and get rid of that structure. Then we have the satellite cells. So in a satellite cell, satellite cells are found in the ganglia. In the ganglia, within the ganglia, we have the neurons. And neurons, a part of their neurons, you find an aggregation of those. Like this is a cell body of neuron. This is a cell body of neuron, and you find in a ganglion a number of these. Okay. And so this is the cell bodies. So when you see several cell bodies surrounded by connective tissue, it's a ganglion. And within the ganglion, you find these cell bodies of neurons, and the cell bodies of neurons are surrounded by these cells. See that the nucleus in black, darker. It is these cells that we call the satellite cells. So they are the supporting cells of the neuron within the ganglion. Please, any question on the neuroglia? Okay. Then let's look at the neuron. We'll look at the neuron. So the neuron is a cell, but it has two main parts. It has two main parts. A part called the cell body, and the other process is called the other parts called processes. So this is a picture of a cartoon of the neuron. That's the cell body, and this is the axon. When it comes, so comment on the neuron cell body. So the neuron cell body is actually the metabolic center of the neuron. And it has an alternative name called the pericardium. And the neuron, the cell body, contains a number of structures that I'll point out. So you have a nucleus and a cell body. A nucleus and a nucleolus. So you can see nucleolus there. So it has a prominent nucleus and a nucleolus. Then also the cell body itself has some cytoskeleton. It has some cellular skeletons in their walls, and that is responsible for providing the cytoskeleton of the neurons. Then the third is the nasal bodies. So nasal bodies are colored substances, and they are formed from the rough endoplasmic reticulum. And they are actually the endoplasmic reticulum. They are formed from the endoplasmic reticulum. When we study the cell bodies, we have an idea of what is happening to the So when we check the cell bodies, then what we are interested in is we want to know what is happening to the endoplasmic reticulum. Then we'll talk about the processes. So the processes, there are two main types of processes. We have the dendrites, then we have the axon. So dendrites are smaller processes. They are smaller processes and they occur numerous. And what they do is that when it comes to the electrical information transfer, it comes to information transfer, this is in a form of electrical impulses. The dendrite will receive the information and transmit it to the cell body. Dendrites are not covered by the myelin sheath. Okay, but in the case of axon, the axon, what it does is that it transfers information. So the axon transmits information in the form of nerve impulses away from the cell body. So it moves it in this direction, but cell body brings it to the So you have the axon transmitting information away from the cell body, whilst the dendrites will bring the information to the cell body. Now, the axon is usually a single process.
Hello.
Yes.
Hello, Doc.
Yes.
Please, let's go to the diagram.
Which diagram?
The neuron.
Yes.
The diagram shown there. Please, you made mention that the neuron is made up of the cell body and that of the processes. And can you clarify this for me? The parts are many there. Can you kindly let me know the parts that are made up, that the cell body is made up of, and then the ones that the processes is made up of?
Okay, so these processes are these extensions, cytoplasmic extensions. Okay, so if you look at the cell body, you see that the cytoplasmic extension is this one and these others. Okay, so it is this, the smaller ones are usually referred to as the dendrites. They are smaller and they occur in large quantities, large numbers. They are called the dendrites. Then this particular process, that emanates from the cell body is the axon. All other parts, when I'm talking about the parts of the axon, I'll add. Is it a little clear?
Okay, so let's go. Maybe in the processor, I will get the whole idea.
Okay. So we have the axon, which is the axon, and it transmits impulses from the cell body. Now, the difference between an axon and dendrite is that axon is thicker in diameter and usually occurs as a single process. So the axon is thicker than the dendrite and it occurs as a single process. And it may be covered by myelin. So you have myelin sheet covering the axon of the neuron. Now, so you have the green, which is the myelin. You have areas, it becomes a myelinated axon. And even in that, there are still segments that are not covered by myelin sheet. They're not covered by myelin sheet, and we call those segments nodes of Ranvier. The nodes of Ranvier. So we have the nodes of Ranvier. There are other processes which may extend. The process that extend from the cell body. This brings us to other parts of the axon. So I will start from bottom. We have what you call the axon hillock. The axon hillock. The axon hillock is the initial segment of the axon. That's the initial segment of the axon, and this is where the action potential is generated. So that is where the action potential is generated. Then we have the axon. When you get to the tail end, the axon starts branching into smaller processes. Then also, so we have the axon. So the axon terminates as various branches, and at the tip of the branches we have what you call the synaptic knobs. The synaptic knobs. Okay. So then we also have other terminal branches. You can have a branch like this. They may not necessarily come from the extreme. They may not necessarily come from the extremity, but they can branch off from the sides of the axon. Okay, and they will all have the bonuses. So they will have the synaptic knobs. They all have the synaptic knobs. So these are what we call the axon collaterals. Axon collaterals. So please, are you able to now follow the diagram, the labeled diagram of the neuron?
Yes, please, I'm following.
Okay. Thank you. Okay. Then we look at the neurons. So having looked at the neurons, we have a structural classification of the neurons. So we have neurons, we can identify the cell body, but we have some different types. So we have a unipolar neuron. We have a unipolar neuron. When we look at the structure, this is a neuron that has no dendrites, but it just has a process, and that process happens to be the axon. So here you have the neuron and you have an axon connecting to it on the slide. Then we have the bipolar neuron, which is this is a bipolar neuron. So you look at the cell body and you notice that two extensions are emanating from the cell body. Usually one will act as an axon, the other will act as a dendrite. Then we have the multipolar neurons. Multipolar neurons has a process, but it also has several branches. So the multipolar neuron has several dendrites and also the axon.
Sir.
Yes.
Please, with regards to the unipolar, when you look at the ends, they seem to look like the dendrites, but you said they do not have dendrites, if I get you right. So what could those structures be called, or how do we name those structures at the ends of the unipolar?
So they are often they are axon terminals. So the dendrites are not coming directly off the cell body. Okay. So it is only a single process coming out from the cell body. So in that case, that single process is the axon. So that neuron doesn't have a dendrite. But when you look at for example, the unipolar, Some of the neurons are some of the axon. This is they are axon. One it branches, one goes down, the other goes up. Okay, but the function is that usually what happens is that one end is connected. One end of the axon is connected to the structure that they want to transmit information. They want to transmit information from. Okay, so and the other end of the axon transmits information to the central nervous system, being it the brain or spinal cord. Please, is it a little clear?
Yeah. But let's move on.
Okay. We have sometimes some unipolar that looks like bipolar. They look like bipolar, and we've given them a term that's a pseudo unipolar. Now, if you look at the diagram which is the unipolar is preferably referred to as a pseudo unipolar because its structure looks more like a bipolar. Okay, looks more of a bipolar. It's not but actually it is a unipolar because a single process comes out from the cell body. Then also neurons can be classified based on function. And when it comes to the function, what happens that we look at whether the neuron is transmitting the information. When I say information, I'm referring to impulses. So we have like sensory neuron, and these are neurons that carry impulses from the receptors, usually the environment, our body. The body has receptors which will detect the changes in the environment. So it is the sensory neurons which will transmit information from these receptors from our body and take it to either the brain or spinal cord. Then we have the motor neuron. So they carry impulses from the spinal cords and bring it to the effector organ, which is either skeletal muscle, smooth muscle, cardiac muscle, or the glands. Then within the CNS, we also have the interneurons. So they serve as a relay. They relay the information from the sensory neuron and give it to the motor neuron. Okay. So these are the different ways of classifying neurons. I don't know whether there are questions.
Yeah, so I'll suggest you bring the inter so that the understanding will be clear. The inter in the middle so that the motor neuron comes. I don't know if that will do. So that says it relays from the inter to motor.
No, okay, in terms of the writing, yeah, there's an interneuron. So connecting them. But this is well, it can be done. It can be done, but I think it may not make any difference. But usually when people are reading, the first point mentioned the sensory neuron, then the second point mentioned the motor neuron. Then the interneuron, you indicate that it's like a relay neuron. It connects the sensory to the motor. So in that case, you would have mentioned them. I just hope that I'm responding to the exact thing that you said.
Oh, it's okay. It's okay. Just that because it links the motor and then the sensory. I was thinking maybe if it should be in the middle so that the understanding will be clear. But it's okay. It's okay.
Okay, so maybe you can add a picture. Maybe a picture will do. Okay.
Hello, Doc.
Yes.
Please let me ask this question in the form of drawing you back. You made mention of the classification of neurons, and
Yes.
according to their functions, and you talked about the sensory, the
Motor
relay,
and
and then
inter.
the motor. Okay. So the first one that you classified, the first type that you made mention of the bi, the uni, and the multi. How did you classify them?
So
Is
that
it
is based
based
on
on the
structure.
number? Based on
It's,
structure.
it's a
Okay.
structure, and it's according to the number of processes coming from the cell body.
Okay. Okay. All right. Thank you.
Okay, sir, please. I also have a question.
Okay.
Sir, please, my question is sometimes let me use the boys. When we are playing football, you may get a cut, but you will not notice it or you won't see it until maybe you are done playing. I want to find out what accounts for that.
Okay, so if you get a cut and you are unable to sense it, then it's likely sometimes the cuts you can, from experience. So if the cut is unable to elicit a marked pain, a pain that is noticeable, it's very difficult for your body to be able to identify that. Sometimes maybe also the cut, if the cut destroys results in the destruction of some sense receptors, then if the receptors are destroyed, then you may not be able to sense it. That is what comes in my mind immediately. I don't know whether
So does it mean at that moment the sensory nerves are not working?
The sensory nerves, well, if one thing is for you to be able to perceive the changes in the environment, you also need receptors. See, you need some things to detect changes in the environment, which we call the receptors. Then they are visually connected to the sensory receptors, the sensory neurons. Then the sensory neurons would also transmit it upwards to the brain for processing and perceiving. As to whether we cannot just say that the neurons are not functioning. We even have situations where people may have trained them, they may be absent-minded. They may be absent-minded, and it is when later they become conscious before they realize that they have actually been hit. Other than that, some people may not even pay attention to those parts of the body. So it's like there are a number of factors that we may need to consider. Okay, in this scenario that you are indicating.
So a follow-up to what my friend is trying to narrate.
Okay.
Sometimes in football, this one I witnessed it where two players crashed and one of them fell down, and we realized that as he was on the ground, he had a broken thigh. The thigh bone was broken, but unfortunately, he didn't identify that he had a broken bone. So, we were trying to lift him up. So as we were trying to lift him up, we realized that he could not stand on his bone. Then that was when we realized that his bone was broken. So my friend was trying to say that at that critical moment, he himself was unable to identify. Probably the nerves were unable to identify that he had a broken bone. But so we were trying to lift him up. We realized that he was not able to stand on his feet. And he is trying to find out what causes that.
Oh, but you answered.
Okay, sir.
So let me add something to what you are saying. You see, when we look at the body function, it's not only the nerves. When I told you about the functions of the nerves, one is perceiving, the other is the responses, and I even added memory. So the response part. So you were able to now detect that your friend had a problem with the bone. When he wanted to now use that function. So when you want to now use that function, that is when the defect will pop up. But when you have not used it and you are there, okay, you are there. For you know, the pain that you are experiencing, it's not. It's maybe some minor pain to you, which your body may ignore. But when you start to use it, then in that case, the nerves are activated. You need to move the muscles. Some of the muscles need to move. And if you are having and they should have an intact bone. But in the course of trying to move, that is when that you can detect the changes. It's a number of factors come into this. You cannot just use a single explanation to explain for all concepts. That is my worry, my challenge with these questions. Okay, so please, I don't know whether those comments will also help or give another dimension to it.
Hello, sir.
Yes.
Let me add to this. There is a condition called delay inflammation. So when delay
Delay
inflammation occurs,
inflammation.
delay inflammation. So sometimes when it occurs, you may get injured seriously, but you will not notice it until some hours later.
Okay.
So I experienced a situation in football where two people crashed and then one broke his leg, but they finished playing the ball, and after the person had come home and he is resting, then his leg began to swell, and later on we realized that he had a broken leg. So it was a delay inflammation which occurred at that instant.
Okay. Yeah, I don't know whether there are any additions. Okay, so
Can you
what?
ask question?
Okay.
So I'm asking if someone is having this ailment, stroke, does it mean that all the nerves is not working at all?
It's not necessarily the nerves which are not working. The body, you see, the body function. It's not only nerves. It's also we should also factor in information transfer within the brain and the spinal cord. So it depends on which part is affected. But if it affects in stroke, it affects the brain, and the brain is the overall boss. So even when you send information to it and it does not interpret it and give you a feedback, it's like nothing is happening. So I hope you get it.
Yes, sir. Yes, sir.
So it's not the nerves. It's just an aspect. It's just an aspect of like the pathway. Mhm. So maybe permit me to add this kind of comment. So I've indicated that, you see, the nervous system. Oh, let me finish this slide, then we can bring in the question. The nervous system is divided into two: central nervous system and the peripheral nervous system. And the central nervous system is made up of the brain and the spinal cord. Now, yeah, the brain and the spinal cord are organized into white matter and gray matter. So the gray matter is usually found at the outer region. The gray matter is found at the outer region. Then the white matter is found in the inner region. So what happens is that the white matter component of the central nervous system is formed by the axons, predominantly by the axons of the neurons. So it is the axons of neurons which come together to form the can we get it muted? So the white matter is the predominantly made up of the axons of neurons. And the gray matter, the gray matter is also, but the gray matter, what you find is that you find predominantly the cell bodies of the neurons. Now, the picture that we showed you is a three-dimensional, but it will interest you to know that, for example, a pseudo-unipolar neuron, we have a pseudo-unipolar neuron, which is starting from, let's say, the spinal cord level, which is at your lumbar region, all the way to even past the neck, all the way to the brain. That tells you how long an axon of a neuron can be. Okay, so the axons within the white matter, you have axons. Oh, let me finish making the point. So the pictures that you are seeing, the structure of the neuron is more like a 2D diagram. But you have to bear in mind that these are three-dimensional structures which are present in our body. Okay, so you can have a neuron whose cell body is outside the spinal cord, but its axon will enter the spinal cord and move all the way up to the brain. Okay, now when you have something like that, you see that when you cut a section of the brain or spinal cord, you will see only that section of the neuron that is passing through, but you may not see the entire neuron. That is one of the things that I want to bring across. Okay, so when we go to the brain and the spinal cord, we can divide it into white matter and gray matter. So the white matter part is formed by axons of neurons. Predominantly, when you look at it under the microscope, you will see predominantly the axons being present. Then in this same brain and spinal cord, there is a region called the gray matter. And that gray matter, predominantly, you will see the cell bodies of the respective neurons. So predominantly, you see the cell bodies of these neurons. Also, in the white matter, occurring in the white matter, is sometimes some gray regions which we call nuclei. Is the gray regions called the nuclei, the gray matter, can either be at the periphery, then the white matter will be at the center, at the core, or sometimes the gray region will be at the core, then the white matter will be at the periphery. But in either case, within the white matter, you may find some gray regions, which we call the nuclei. Okay. Now, in this nuclei, it is the cell bodies which are predominant. You find cell bodies predominantly in this gray regions called nuclei. Okay. Then we come to the peripheral nervous system. We have nerves and ganglia, but these are outside the brain and the spinal cord. So you can see some nerves here. You see nerves, and occurring among them is the ganglia. They are swelling. Now, nerves are formed by axons of neurons. So it is the axons of neurons which come together to form the nerves. Then we have ganglia, which is also a structure outside the brain and spinal cord, but it is actually a collection of the cell bodies of neurons. The cell bodies of neurons. So these are occurring among tissues and also in between nerves. Okay, but it is made predominantly of the cell bodies of neurons. So this brings me to the first part where I said that a nervous tissue, when you take a nervous tissue, anytime you take a nervous tissue to look at it under the microscope, you should be expecting a part of the neuron and the neuroglia. The part of the neuron that you see may be the neuroglia. Maybe either the cell body or the axon. Okay, then it occurs with the neuroglia. Please, so I'll pause, and if there are questions, you can ask me. Okay. Now, I've commented on. Oh, okay. A question.
So I was asking that in case if some of the brain cells get damaged,
yes,
we get a replacement, or is damaged forever?
Okay, the regeneration is so slow. It's not fast. It's very, very slow that it can even take a decade. Can take more than a decade. So nowadays, yeah, it's very slow. So for it to, for the recovery, it takes very, very long time. So what we advocate is that you rather prevent it from happening.
Thank you.
Okay.
What causes the cardiac muscle to be weakened?
Please, a question.
What causes the cardiac muscle to be weak?
The cardiac muscle to be weak.
Yes, please.
As in myocardiac infarction.
Yes, please.
Myocardiac infarction, the cells are dead though. So usually when they lose the common cause is when they lose the blood supply. When the oxygen and the nutrients that should reach them for them to function is cut off, then they are liable to die.
So is that the reason why there is this condition called cardiomyopathy? And is that condition life-threatening?
Please, the second part of your comment.
I'm asking whether that accounts for the reason why cardiomyopathy results in cardiomyopathy.
Yes. So when the blood supply is usually that's the most common cause when it is cut off. Oxygen and nutrients is cut off, and it will result in cardiomyopathy.
I'm asking if cardiomyopathy is life-threatening.
Whether it's life-threatening?
Yes, please.
Hey, it is. It is life-threatening because of the function of the heart. The heart is supposed to pump blood throughout the body, and if your pump is not, if your pump is unable to move blood through all the body parts, your body parts will starve. Look at the brain, including the brain. The brain is also supposed to receive the blood supply, though the blood is not supposed to come into contact with the brain tissue, but it still needs to reach the blood-brain barrier so that the available nutrients that the brain can use can also, at least it can reach them. So it can, by diffusion, they can reach them. But if because of cardiomyopathy, we don't have enough blood reaching the brain, it will also result in stroke. Yeah, so the brain cells will die, and it will, yes. So to me, it's life-threatening.
yourself.
Yeah, please, a question.
So please, I'm asking whether there is something that could be done to restore the the cardiac muscle to its original state?
Well, I think okay, I am not in that position to be able to give you the answer. So this will be the expert. I'm not an expert in that area, but
Okay, thank you, sir.
give information. Yeah. Okay. Now, when I was talking about when at the early part of the lecture, I was talking about information transfer, and that information transfer is what we call the impulse. That is a nerve impulse. So the remaining part will be looking at how the neuron generates impulse and transmit it to the through the nervous system. So that is what we are going to look at. So the first thing that I want you to understand is that when it comes to the plasma membrane of a neuron, just like that of a muscle cell, the plasma membrane of the neuron is electrically polarized. So for many cell membranes, they are electrically polarized. And when they are electrically polarized, what usually means is that we have the outer layer being positive, electrically positive, then the inner layer being electrically negative. So the key thing is that the cell membrane of a neuron, just like every other neuron, you have the plasma membrane. The part of the membrane, this is too. So you have the plasma membrane of the neuron. The cell membrane is electrically polarized. So it has more negative charges on the inner aspects. Then it has more positive charges on the outer aspects. Now, and this is because the electrical distribution of the ion distribution are imbalanced. So you end up having more positive charges going towards the outer part and relatively more negative charges coming to the inner part. In that state, we say that the plasma membrane is in the resting state. So we say that the plasma membrane is in its resting state. And because of the charges of the two layers, it can generate what we call a potential difference. It's just like our electricity. You have positive terminal, negative terminal, you generate electric current, electric voltage. And the voltage that exists across the plasma membrane is what we call a membrane potential. Membrane potential. So to comment on the membrane potential, one thing is that there are more negative charges on the internal aspects. More negative charges on the internal aspect of the plasma membrane. Then there are more positive charges on the external aspect of the plasma membrane. And this, the polarity, one layer becoming more positive, then the other layer becoming more negative, at least is caused by the differences in the rate of diffusion of sodium and potassium ions. So in the plasma membrane, sodium ions are moving across the plasma membrane, and potassium ions are also moving across the plasma membrane. So this is attributed to sodium and potassium. So usually within a very short time, within a time, there are some designated ion channels for potassium or sodium which may open. So there may be designated ions that will transport the sodium and put at least the sodium and potassium, okay, to the other side. Okay, so one, in the body, in the plasma membrane of the neuron, there is more transportation of potassium ions outside the neurons. So we have, this is the cytoplasm, and that is the ECF, extracellular fluid. So there is more movement of potassium ions to the outside of the neuron. Then we also have transport of fewer sodium ions into inside the neuron. So we don't also have, there is more potassium ions going outside, and there is very few sodium ions coming into the cell. So what it means is that the sodium, which is on the external part of the cell, may even add up to the cells, the potassium ions in the outer part of the cell, and there will be a lot. So the outer layer will be more positive, then the inner layer will be relatively, will be less positive, or you can say negative. Now, this state of the plasma membrane.
Sir,
yes.
Can we have instances where the negative dominates the positive? And in that case, what happens?
Okay, so I will come to that. So we have a situation where it can be reversed. So I will talk about that in a jiffy.
Okay.
Now, this state where we have a plasma membrane having the outer layer being positive, inner layer being negative, is actually stabilized by one of the proteins. We call it the sodium potassium ATPase pump. Now, this pump ends up adding more positive charges to the outer layer of the cell than the inner layer. So, for example, it pumps 3 sodium ions out of the cell onto the plasma membrane, but 2 potassium ions into the cell. So when you also have more positive ions being added to the outer layer of the cell, it makes the outer layer more positive.
Sir, please, with all due respect, it seems I'm not getting where the negative charges are coming from, because at the beginning you said that potassium ion is found at the outer layer, but since you are saying negative charge, I'm not getting the concept.
Okay, so when it comes to the body function, let's say like we have 3 and 2, the numbers 3 and 2, positive 3 and positive 2. We know that positive 3 is more positive than positive 2, right?
Yes.
Yes, but which one is more negative, positive 3 or positive 2?
Positive 2.
Exactly. So when it comes to, so if you noted, if you have paid attention to what I've been saying, I've been using the word relatively.
Okay.
So when we say relatively, the outer layer is relatively positive. The inner layer is what? Relatively negative. So that means that there will be more positive charges on the outer layer than the inner layer. So when there are more positive charges on the outer layer than the inner layer, if you want to use polarity, you say that the outer layer is positive, the inner layer is negative. I don't know whether this statement also makes it a little less cumbersome.
It's clear now. I was not getting where the negative is coming from.
Okay.
Since in the beginning, you stated it's positive.
Yeah, so the outer is they are relative charges. Okay. Thank you very much for the question. So you have this potassium ATPase, sodium potassium ATPase, adding more positive charges to the outer layer, but fewer positive charges to the inner layer. And this causes the outer layer to be more positive than the inner layer. So these things happen so that the cell membrane will have an relatively outer positive layer and relatively negative positive layer. Now, it gets to a time that when the neuron is stimulated, the membrane potential across the plasma membrane can change. The membrane potential across the plasma membrane will change. And when it changes, what happens is that instead of the outer layer being to stay as positive and the inner layer to stay as negative, it will rather reverse it so that the inner layer will be relatively more positive than the outer layer will be negative. And when that happens, we use a term, we have depolarized the membrane. So the polarity is that the polarized state is that outer layer should be relatively positive, inner layer should be relatively negative. But when you depolarize it, you have reversed it. So the outer layer will rather become relatively negative and the inner layer will become relatively positive.
So in that case,
sorry,
in that case, are we going to have more more of the positives inside and then the negatives outside?
Exactly.
In terms of the movement, I just wanted you to explain like the movement. So in that case, you have fewer positives or more positives entering the membrane and then fewer negatives moving out of the membrane.
Or fewer positive going to the membrane, outer part of the membrane. So at the end of the day, there should be more positives on the inner membrane. Okay. Then in terms of positives, the inner membrane will have more positive charges than the inner membrane and than the outer membrane. Okay. Or if we are even supposed to talk about in terms of negatives, we want the inner membrane to be positive. What it means is that there should be fewer negative ions which should go to the inner membrane, but there should be more negative ions which should go to the outer membrane.
Yes.
So yes. So to achieve this state, that is the depolarized state, this is actually caused by what we call the influx of sodium ions. Influx of sodium ions. When we say influx, we are looking at inward movement. So movement from the ECF to the ICF. From the ECF to the cytoplasm, which is the ICF. So when a neuron is stimulated, there is an inward movement of sodium ions, a lot, from the external part, that's the external extracellular fluid, ECF, all the way to the ICF. So that makes the inner layer more positive than the outer layer. And one thing is that these ions are passing through some channels, which we call the sodium-gated channels. I'm thinking that maybe we may, I'll discuss with the other group. Okay, let's finish and let me ask my question. Then also, when the neuron is stimulated, there is also another possibility where the inner layer can rather become more negative than the outer layer. So for it to happen, what it means is that there should be more, the inner layer becoming more negative than the outer layer. It is the possibility is that there will be more negative charges coming to the inner layer and fewer negative charges going into the outer layer, or there should be more positive charges leaving the inner layer so that more positive charges reaching the outer layer. Now, when it happens like that, we use the word hyperpolarized. So the membrane has been hyperpolarized. And there is, in the lab, there is a graph, a machine that is made to read the potential across the plasma membrane so that we can be able to monitor the changes in the membrane potential. So if you look at this graph, usually when the one thing is that the potential differences, the voltages across the plasma membranes are usually in the negatives. That is, they are mostly in the negatives. And so if it becomes less negative, then it means that it has become relatively positive. And if it becomes more positive, so that is to draw your attention to that fact. So you have this machine, the output, which monitors the plasma membrane. So here you notice that on this output, we have 0. And as you move more down, more vertically down, you are moving towards more negative. And as you move more up, you are moving towards relatively positive. So we have situations where the membrane, when the neuron is stimulated, the membrane can become relatively less negative, or you can say more positive. Then in the second picture, you notice that the line graph dipped. So in that case, the membrane potential becomes more negative. So the first instance is a depolarization, and the second example is for hyperpolarization. Please, any question? Okay, maybe I can continue. Now, the hyperpolarization can be caused by the efflux of potassium ions. So when we say efflux, we are looking at the movement of ions outside the plasma membrane. Please just give me some few seconds. Let me sort out something. I think for some people, when there are two diagrams, they get they are unable to follow. So okay, let's move on. So the plasma membrane is polarized. When you stimulate it, it can be depolarized, or when you stimulate it, it can be hyperpolarized. That is the point that I have made. Okay, and one thing is that when it is depolarized, after some time, it tries to revert to the original state, which is the polarized state. So here you follow the graph, you see that when it is stimulated, the membrane potential becomes less negative or more positive. Then after some time, it comes back to the original state. That's the resting potential. And in the second example, after stimulation, the membrane potential became more negative. But after some time, it went back to the original state. So it is something that it changes is dynamic. Okay.
Now,
yes.
The resting potential, in a simple sense, could mean an equilibrium state of the positive and the negative, if I get you.
Exactly. Yeah.
Okay.
Yes. Now, there is also something that I will have to add to the membrane, changes in the membrane potential. Now, with the neuron, the plasma membrane, when there is depolarization, it has two outcomes. And the outcome is dependent on whether a threshold value is reached or not. And this rather gives rise to two types of depolarized membrane potential. We have what you call the graded potential and action potential. Graded potential and action potential. So the graded potential is generated when the depolarization of the membrane does not reach a threshold value. So depolarization is when the plasma membrane potential becomes less negative or more positive. Okay, but even whilst it becomes more positive, there should be a limit. If it does not reach a particular threshold, we will still have what we call a graded potential. Now, when you have such a condition, after some time, the membrane potential will just die down. It affects a number of parts of the neuron, the plasma membrane. Then it reverts back to the resting state. That's a resting potential. So that information that impulse, it's also a form of impulse. It can just travel very on short distance, then it fades away. Then there is another kind of potential, which when the depolarization of the membrane occurs, it takes the depolarization to the threshold value. So when the membrane depolarization reaches the threshold value, it triggers another a series of activities, which we call the action potential. So when the depolarization reaches a threshold value, it triggers a series of activities, which we call the action potential. And what this makes this action potential, we classify it as either an all or none response. It only comes in when the depolarization reaches a threshold. If the depolarization does not reach a threshold, action potential will never occur. But if the depolarization of the membrane reaches a threshold, then action potential will occur. Okay, so the action potential is able to travel over long distance on the axons, and that is what constitutes a nerve impulse. So when the action potential is generated in a neuron, the action potential will be conducted along the axons of the neuron as a nerve impulse. Okay. Now the action potential occurs at the axon hillock of a neuron. It occurs at the axon hillock of a neuron. And when it is at the axon hillock that when the threshold has been reached, then you have an action potential being generated. So in this diagram, you see the cell body of a neuron, and that is the axon hillock. So it's the depolarization reaches the trigger zone, which is the axon hillock. It meets the potential, then it meets the threshold, then the action potential is generated, which is conducted along the axon. But in the second picture, the depolarization reached the trigger zone or the axon hillock, and it didn't reach the threshold, so no action potential was generated. The action potential, when on the machine, when we monitor the action potential according to the changes in the potential, just like we monitor the depolarization, the action potential has a stereotypical shape. So it has different phases. So this is how it looks like. So in this diagram, the neuron has a cell membrane which was in a resting state. Then they brought in a stimulus, something to stimulate the neuron. Then the membrane potential depolarized. So when it depolarizes, it becomes less negative or more positive. So when it became less negative, it rose to this, but it was able to meet the threshold, and immediately the threshold is reached, then the action potential would occur. Action potential is made up of phases. We have the first phase, which is called the depolarization phase. The second phase, which is called the repolarization phase. And the last phase, which is called the we have a hyperpolarization phase. Then finally, a repolarization phase. When we say repolarize, repolarization is a resetting. So after depolarization, if the membrane potential wants to come back to the original state, it is repolarized. When it is also hyperpolarization and the membrane potential wants to come back to the original state, it is also still repolarization. So the action potential is made up of the phases. Immediately the threshold is reached, then the action potential will set in. But the action potential is made up of a number of phases. There are 4 of them. So we have the depolarization, repolarization, then we have hyperpolarization, then we now have another repolarization phase.
Sir,
yes.
Can this be related, this action be related to, let's say, someone who smokes weed or a drunkard, and therefore the changes that actually occurs in such people can be related to the changes we are seeing in this particular instance of our lesson? If you can actually relate this, or can it be related in the first place? And if it can, how do we relate this depolarization, polarization, the threshold, the repolarization, and etc. to
Please,
such a test?
the question again.
Okay, so sir, I'm saying that in one, can we relate this process or these processes to when one takes in weed and the behavior, the change in behavior of the person, or probably, let's say, a stimulant, a depressant such as alcohol, when alcohol is taken in. Can we relate the behavior of the person to what we are studying over here, and how?
Okay, we can relate it. It's just that all these I'm referring it to the to build my point and explain to you what happens on the axon. But we also have another part which is called the synapse. And most of these the drugs, these recreational drugs, alcohol and the others, they have serious effects on the other synapse. They have serious effects on the synapse. And based on what happens at the synapse, it can also affect the nerve transmission. Okay, just that most of them will have effect on the synapse, at the synapse, which we have not reached there yet. Please, I hope you get me.
Yes, sir.
Okay. So the action potential. So for example, I think I've already commented on some of the ionic bases. The dipolar. Okay, so the ionic resting potential. Usually, you have more positive charges reaching the outer layer, less positive charges reaching the inner layer. And it is because the potassium ions are moving out of the cell. Okay. Then we come to the depolarization phase. And the depolarization phase, you have influx of sodium ions. So sodium ions rush inward the cell and make the inner layer more positive, and the outer layer become relatively more negative. Then it gets to a point that the thing is reversed. So we go to the repolarization phase. And in the repolarization phase, it has to do with the membrane polarity trying to be reestablished. And in the repolarization phase, potassium ions then also start, in depolarization phase, the inner is positive, outer is negative. In the repolarization phase, the first repolarization phase, positive ions now start leaving the plasma membrane. It leaves the cell to the outer layer. So you have more efflux of potassium ions moving to the outer layer of the plasma membrane. And this process continues for a long time, that by the time the plasma membrane realizes, there will be more positive, which have like left the inner aspect of the plasma membrane. More positive charges have left the inner layer of the plasma membrane, so that the inner layer is now more negative or less positive. So when it becomes even less positive, then you find the membrane potential dipping. The membrane potential now dips. Then finally, we have another instance where the sodium potassium ATPase pump, that pump which stabilizes the resting membrane potential, will act to bring back the membrane potential to the resting state. Okay, so that electrical impulse that is being transmitted along the axon of a neuron is actually an action potential. So it is action potential which is being transmitted along the axon, and that is what presents as a nerve impulse. What I would want to add, I think that I have excluded on the slide, is the fact that the sodium potassium ATPase pump resets the plasma, the membrane potential to its resting state. Okay. I will add it. My computer is misbehaving. Okay, please, any question? Okay. So we have what you call a refractory period. We have what you call refractive period. And the refractory period refers to the fact that, you see, a neuron not generates an action potential. When you activate, you generate an action potential. Before the neuron can generate another one, there should be some time, some period. Please, a moment. Okay. So when it comes to the action potential, when an action potential is activated, the next one can only be generated after some time. So it doesn't matter the strength of your stimulation. It can only come in after some time. And that is when we call the time difference. That is the time difference that we call the refractory period. So the period between two stimulations during which the nerve fails to respond to a stimulus of threshold intensity. Okay, and this refractory period is very, very important. It helps information transfer in one direction. So this will bring me to the transmission of nerve impulse along an axon. So a nerve impulse is generated when action potentials are conducted along the axon. And our nerve impulse, which is actually action potential, which is being transmitted along the axon, it should travel in one direction. It should travel in one direction. And for the neuron to ensure that, what happens is that if this is the neuron and this is a region, that's the axon hillock. This is the axon hillock because of the expanded region. So when an action potential is activated, okay, and the action potential in this picture, the action potential is demonstrated by the active regions. Usually, the region of the axon closer to the cell body is in refractory state. And the region is far from the cell body is in an inactive state. So because the region that is closer to the cell body is in a refractory state, it cannot generate an action potential, but rather it is the inactive region that can generate the action potential. So when you have an active region, the next region that can also generate an action potential will be a part of the axon, which is far from the cell body. And when also this region become active, this active region will rather be refractory. It cannot generate an action potential again. It will only be the region which is far distal to this region that can generate an action potential. So because of refractory period, then action potentials will be transmitted along the axon in one direction, and it would be towards the axon terminal. Please, any question? Okay. Now, in the nervous system, information cannot be, for information to reach, to move from the external environment to the brain, it will require more than one neuron. And it will be necessary that the neuron will transfer the information. The neuron will transfer the information to another neuron. And the point at which the two neurons make contact, the neurons will make contact for that information transfer, we call it a synapse. And usually it is a gap, a small gap that lies between the two neurons. So a small gap between one neuron and another neuron or an effector cell is called a synapse. And that is where the information transfer occurs. That is that transfer of impulse occurs. Now, usually when you have a synapse, we have the neuron that comes before the synapse. We call it the presynaptic neuron. And the neuron that comes after the synapse, we call it the postsynaptic neuron. So this is the synapse. So we have a presynaptic neuron and a postsynaptic neuron. In our body, most types of the synapse is a chemical synapse. So the chemical synapses are specialized to allow the release of chemicals called neurotransmitters. So neurotransmitters are actually released from the presynaptic neuron into the synapse so that the chemical will interact with the plasma membrane of the postsynaptic neuron. And the chemical synapse is made up of two parts. So we have the axon terminals, the knobs of the axon terminals. Then we have on the postsynaptic membrane, we have the neurotransmitter receptor region. So in the presynaptic, in the axon, the knobs of the axon terminals, we have synaptic vesicles and they store neurotransmitters. That is in the presynaptic membrane. Then in the postsynaptic membrane, it has this receptor region, which will receive the neurotransmitter. And these two are separated by a space, which we call the synapse, or we can say the synaptic cleft. So there are a number of steps which would occur in order to allow for information transfer. So based on what I've explained, you have action potential arriving at the presynapse, the axon terminal, and there is influx of calcium. Calcium moves from the external into the cytoplasm. So calcium ions will move. Immediately the action potential arrive at the knob, calcium ions will move inward in the axon knob. And when the calcium ions move inward, what happens is that it will merge or fuse with the synaptic vesicles, and it will cause the synaptic vesicles to move towards the synapse, the membrane. When it gets into the membrane, the synaptic vesicles will release their content by exocytosis so that the neurotransmitter will be released into the synaptic cleft. The neurotransmitter will bind to specific receptors on the postsynaptic membrane. And the binding of the neurotransmitter on the receptors often would open ion channels, and that can generate a graded potential. So when the graded potential reach the axon hillock, and the axon hillock it's able to reach a threshold, then the action potential will be generated in the postsynaptic neuron. Finally, we'll look at how are the neurotransmitter effects terminated. So there are a number of factors, like, for example, there will be a reuptake of the neurotransmitters. So it's like the neurotransmitters will be sucked out of the synaptic cleft. So there is reuptake of the neurotransmitters back into the presynaptic terminal. Sometimes there are enzymes in the synaptic cleft that can break down the neurotransmitter, or sometimes the body will just allow it to just diffuse away so that its effect will reduce. We have factors that affect the movement of action potential across the axon. So if action potentials supposed to be traveling along the axon at a faster pace or a slower pace, there are a number of factors that can affect that. So one of them is the myelin sheath. When we have myelin sheath on the axon, it facilitates the transmission of nerve impulses, the action potential. So the action potential will be transmitted very at a faster rate. So for example, in this picture, you will see a neuron, one neuron is myelinated, the other neuron is not myelinated. And you see that the electrical current on this is able to reach the axon terminal at a faster rate compared to this other neuron, which is not having any myelin on it. Another factor is the size of the nerve. If the neuron is having a bigger diameter, the action potential will travel at a faster pace. If it's having a smaller diameter, action potential will travel at a slower pace. Then the other one has to do with temperature. Higher temperature will also facilitate the movement of the ions. So it helps the action potential to be transmitted at a faster pace. So this is what we have for you so far as the nervous tissue is concerned. So far as the nervous tissue is concerned, it's a two-part. We started by telling you the structures in the nervous tissue. So I commented on the neuroglia. Then I came to the neuron, the types of neuron, and now took you through how the impulses, that what we call a nerve impulse, that electricity that is being transmitted along the axons, how they are generated. So I gave the basis as starting with the resting membrane potential and establishing the fact that the membrane potential can change. And when it changes, it can either be depolarization, we can also have hyperpolarization, and there is also another one, repolarization. So if it is hyperpolarization and it is coming back to the resting state, it is repolarization. And if it is depolarization and it is coming back to the resting state, it's still repolarization. Then I indicated that membrane potentials can either be of two. So, but the whole idea is whether the depolarization will reach a threshold potential or not. If it does not reach a threshold potential, that membrane potential will die down with time. And that is what we call the graded potential. But immediately the membrane potential reaches the threshold, then the action potential will set in. And an action potential is a response which has 4 phases. So it has a number of phases, and these things are occurring so fast. Okay, it occurs so fast. Now, when this action potential is being transmitted, so it's occurring along the axon, and it is moving downwards. That is what we describe it as a nerve impulse. And I established the fact that I mentioned the fact that for action potential to be transmitted in one direction, what happens is that when the action potential is happening here, the region that is closer to the cell body is in a refractory period. So it cannot generate another action potential. The action potential can only be generated by the inactive region. It can only be generated by the inactive region. So when the inactive region now becomes active, the inactive region will now be lying here, this down to it. So then the action, that is where it can elicit another action potential. So the action potential will be transmitted until we get to the synapse. And when we come to the synapse, I mentioned that the most of the synapses in the body are chemical synapse. So it requires that the chemicals will help in transmitting, transferring the nerve impulse from one neuron to another. And the name of the chemical is a neurotransmitter. That is the neurotransmitter. Okay. So I indicated the steps that are involved in the transmission of the impulse. So an action potential will reach the knob of the axon terminal. Calcium will move from the external to the internal, and it will merge with the vesicles. Then the vesicles would release their neurotransmitters onto the postsynaptic membrane. In the postsynaptic membrane are receptors, which it will going to interact with. Okay, so I've also indicated how they are being terminated. Okay, then I added the factors that can affect transmission of nerve impulse. So myelin sheath. When we have myelin sheath, it accelerates the transfer. Then we can also talk about the diameter. The thicker the nerve, the thicker, the faster the rate of action potential. Then also I added that the temperature also affects it. So high temperature also promotes faster transmission of action potential, or you can see the faster transmission of nerve impulse. So on this note, I will pause, and if there are any clarifications. Okay, so hey, when are you writing your quiz?
24th.
Yeah.
24th January.
24th of January.
23rd, 24th.
23rd is on Friday.
But what I want to understand is it in January?
Yes, please.
Yes, sir. Okay. And when do you write your second quiz?
After the semester exam.
Oh, after the semester exam.
Yes.
Yeah.
So maybe we should agree on the topics. So you've done, I think this is the 7th lecture. You've done 7 topics. And I think maybe the first quiz, we can let it, I'm thinking that maybe connective tissue, so epithelial and connective tissue and the others can be, and the first 3 lectures can be your first quiz. What do you think?
Please come again.
I'm thinking that the first 5 topics can be your first quiz. So up to connective tissue.
Which are the epithelial tissues?
Epithelium and the connective tissue. So up to the connective tissue can be your first quiz.
Okay, sir. Okay.
Only epithelial and connective tissue.
Hey, but there were 3 other topics.
So from cells to staining to microscope, epithelium and connective.
Yes.
Yes.
I think from cell to connective tissue, that is what he's trying to say.
Yes, is it fine?
Yes, it is fine.
So make it forward.
If I make it 4, then it is epithelial tissue.
Sure.
But how about the exam?
The exam will cover all the topics. That one you will prepare.
Let the lecturer talk.
Sir, please talk.
Please, whoever is recording, please, we have
Return the
finished
ticket
the
for you,
lecture.
please.
I wish you can pause the recording so that these don't fester into that. It's not included in the recording.
Yes, sir. Yes, sir. Very necessary.
So, yes. So, so you are thinking of me as excluding the connective tissue. So that it will be only on the epithelium. Is that the suggestion?
That's
so
my
whatever
suggestion,
you agree, you comply.
Well, okay. So I will send your comment. We will discuss it as a group, as a panel. So with the other course tutors, then I will ask them and give you a feedback through Madam Rebecca. Okay. So as to whether we should cover up to the epithelial tissue so that the others, you come and face them in the exam, or we add the connective tissue. So I'll get back to you. Who will contact me?
First will contact you.
Okay,
He's
so yeah, so my Festus and we'll communicate with Madam Rebecca. Communicate with Madam Rebecca and we'll note the scope. Latest by this week, we should be able to know the scope. Okay, so we'll be left with
this slide. Please, this slide, you don't have it also. Please send it to us.
Oh yes, I'll send it to you.
Okay, thank you.
I'll send the slide to you.
So, Doc, please, are we done for the block? For this
No, we are left with one more topic. The plant.
Okay.
We finished with the animal tissue. So we have one last lecture. We have which is the plant tissue. So we look at the plant. Yeah, so so we will look at it, whether to include the connective tissue or not. Okay. But I think that sometimes, okay, so it's okay. Here, you also want to get relatively good marks. So I understand. So I will get back to you on this one.
Yes,
okay, so on this note, maybe we can end and we meet God willing next week. Okay, but we'll communicate before next week. I trust God that we will communicate and settle on the quiz questions.
Okay, sir.
Thank you very much.
Hmm. Hmm. Hmm.